Hi, my name is Mareike Janiak. I am a postdoc at the University of Salford in Manchester, and I just started this position in the fall. So I'm mostly presenting an overview of what we're working on. I don't actually have results, but I'm hoping to get some useful feedback, maybe, and ideas from people. So this is just a brief overview. And are they advancing the slides?
Nope. I'm still seeing. Yeah, no, it is. Okay. Yeah.
So I'm working on this project. It's part of a NERC-funded project called Rise of the Continent of the Monkeys. And this is in collaboration with Robin Beck, John Mobley, and Ian Goodhead, who are all PIs on the grants. And then Doreen DeFries is another postdoc that started at the same time as me. And basically what our goal is for this grant is to use both fossil data and genomic data to investigate the evolutionary history of an adaptive radiation of primates, the New World primates. So Doreen is in charge of the fossils and I'm in charge of the genomic data. So New World primates are also called platyrrhines, and they are really a textbook example of an adaptive radiation. They're the products of what is thought to be a single founder event around 32 to 35 million years ago, when an ancestor of these primates rafted across the Atlantic from Africa. And this hypothesis used to be really controversial, but we actually have pretty good evidence that this is what happened. And we also have this evidence for other taxa, so marsupials, for example, and there's also evidence that the same thing has happened with lizards, just the other way around. So it's a very cool system because of it's this adaptive radiation that just arrived in the Americas and then rapidly diversified to fill a lot of available niches to roughly around 100 species today. So we have a lot of dietary variation within this radiation too, which is part of the project. But for this phylogenetic portion, sorry, I don't know if the slides are actually advancing. Okay, now they are. So there's still things about the phylogeny of the New World primates that are controversial and partly hotly debated. So for example, the placement of Aeotis, the owl monkeys, has never really been resolved, as well as the timing of all of these splits has not been resolved. And there's some evidence that there's a lot of cryptic speciation happening. And this is partially driven by the formation of riverine barriers. So the riverine barrier hypothesis goes back to Alfred Russel Wallace, who, when he was collecting samples in the Amazon, realized that a lot of species occurred on one side of a river, but didn't occur on the other side, or closely related species would occur on either side of the river, just a few kilometers apart, but just, the Amazon was dividing these different morphs of a species. So he proposed this idea that the really high species diversity that we see in the Amazon is due to the formation of these riverine barriers that lead to allopatric speciation. So primates are a really good system to test this hypothesis because they're distributed all across the Amazon. But up until now, we actually had very little genomic data for this group of primates, and most phylogenies that exist are largely based on mitochondrial data, and in some cases just cytochrome B. So for our project, we're using a large collection of samples that John Mobley, one of the PIs on the grant, has collected over the last 20 years. And we're working with collaborators at UPF in Barcelona, Tomás Marquez, and Illumina, who are generating a lot of the sequencing data for us. So we have sequencing data to 30x coverage for 205 individuals at this point, and more to come. And our samples cover almost all the extant genera, except for one at the moment, but we're hoping to get that last genus too. So our goals are threefold. There's a lot more other stuff going on, but for the purposes of this talk, we're interested in producing a fossil calibrated time tree. We're interested in species delimitation, like the number of species that exist for New World primates is kind of unclear. We really don't know, and it's something that's really important for conservation efforts. And then we also want to investigate the blood-brain barrier hypothesis. So for the phylogenetics side of things, we kind of have to parse down all this massive amount of genomic data we have. So I've assembled and annotated complete mitochondrial genomes for all of our samples using the program MitoFinder. And then we've also pulled out 3,000 UCEs using a combined approach of HRAM and phylogeny. And just a very, very preliminary result is that the mitochondrial tree based on full mitochondrial genomes from these samples resolves almost all the groups very well, 100% bootstrap support, except for the problematic iotas. So the owl monkeys remain problematic, and we're hoping to use some of the other data to hopefully resolve this, and possibly some morphological evidence as well. Thanks.